SAIAB seminar this morning. Before we begin, just a couple of house rules. Please make sure that your phones are on silent. Again, for our online attendees, can you stay on mute? And please feel free to drop questions into the chat function or the Q&A function throughout the talk, and we'll get to those at the end of the presentation. So with that, it gives me great pleasure today to introduce Dr. Jessica Glass, who I first met, I think, back in 2012 or 2013. Jess actually helped me set up my acoustic array in the Kawi estuary for my PhD. So it's 11 years ago now. So yeah, we've actually known each other for quite a long time. Dr. Jess Glass is an assistant professor in the University of Alaska Fairbanks College of Fisheries and Ocean Sciences. Her research integrates genomic tools and environmental datasets to address questions spanning the field of fisheries, evolutionary biology, and ecology. Dr. Glass received a BS in ecology and evolutionary biology from Yale University, a master's in fisheries from the University of Alaska Fairbanks, and then returned to Yale for a PhD, where she studied the evolution of a group of fishes, including the jacks, remoras, mahi-mahi, but we know them as dorado, and billfish. She was a recipient of a National Science Foundation Graduate Research Fellowship, an NSF Doctoral Dissertation Improvement Grant, and a USAID Research and Innovation Fellowship. So really impressive CV and lots of grants behind your name. And for the last 12 years, Dr. Glass has collaborated with scientists and researchers from SAIAB, and then became an official honorary research associate in 2021. Dr. Glass is also a tribal member of the Wyandot Nation of Kansas, and she grew up on the traditional homelands of the Peoria tribe in Illinois, and began her position in Alaska in 2021. So with that, I'll hand over to you.
Great. Yeah, thank you so much, Taryn, and good morning, everyone. Thanks so much for tuning in. I want to just say how great it is to be back here. I can't believe it's been 3 years. Let's see if I can get rid of that. Hold on. 3 years since I was here, and in academia, oftentimes we're moving around all the time, right? You're unsettled, and at least in the States, you're moving from institution to institution, and it's so nice to be back in an institution that really does feel like home to me. I started coming here in 2012, as Taryn mentioned, working with, oh, perfect, that's better. Yeah, Gavin and Paul Cowley, and then times were different back in 2021. And I have to say that today actually marks the exactly 3 years to the day that I started my position. I started remotely here in SAIAB because of our friend COVID. And every year I've been taking a selfie on that anniversary, partially to count how much gray hair I'm accumulating. But I was hoping that today you guys could be in the background of my selfie from my work anniversary. So if you don't mind, I'm gonna stand over here and take a selfie. So smile. Awesome. Thank you. It's really it's great to be back. And I say I have this because coming to SAIAB has shaped my graduate education from master's to PhD to postdoc, where I was here full time. And even now, just thinking about doing, the ability of work that we're able to do in Africa and the Western Indian Ocean, and how that shaped the way that I apply my research tools in Alaska as well. But anyway, back to fish. As you know, in the tree of life, fishes are the most diverse group of vertebrates, far outnumbering mammals, birds, reptiles, with over 36,000 species. And so studying the evolution of fishes is important for understanding, how and why there are so many species that are able to inhabit Earth at one time, and so well adapted to their environment, and possibly well adapted to change or maladapted to change. And it also helps us understand the evolution of humans and of vertebrates more broadly, given that, phylogenetically speaking, humans are fish. And as an evolutionary biologist who studies fish in a fisheries department, my work looks at the intersection of this is automated, wow, genotype, phenotype, behavior, and environment. And using that intersection of these components to look at fisheries management and conservation. And so within the intersection of these components, my research addresses several themes like biodiversity, connectivity, adaptation, And the trajectory of this work has aligned with the ubiquity of big data, like genomic data, transcriptomic data, open source environmental data sets, community science, and Alaska Indigenous science. And using these data or these tools to address these themes allows my lab to look at fisheries management and conservation questions like population structure, the distribution of species and populations, as well as the impact of ecosystem stressors, whether those are human or environmentally induced, on populations and species. This is just a snapshot of my lab in Alaska. It's growing very fast. I'm really proud of everyone that is, I say we, and this is what I mean. I mean my students, my graduate students, let's see if this works, or postgraduate students, you would say here, my undergrads, my postdocs, my lab manager. We're actually growing a lot. I'm doubling the number of postgraduate students, adding another postdoc fellow this August. So it's really fun to kind of build that community in a new place, and echoing a lot from what I've learned and gained from how we interact here at SAIAB. And this is our lab mission statement. So we integrate impactful genomics research and bioinformatic tools for the benefit of coastal communities and kin. And I say kin in an indigenous framework of non-human animals and plants and kin that many indigenous peoples feel are closely related, or are closely related to us. And in Alaska, we have over 229 tribal communities. Some of those, their language groups are more divergent than English is to Japanese. So it's a really interesting place to think about the intersection of evolution and conservation and fisheries in an indigenous framework. And I don't have a lot of time to go into this component of my work, but we're doing a lot of work on indigenous data sovereignty and kind of some of the ethics about generating genomic data sets. But for the most part, I've sort of collapsed a lot of what we do into three kind of core questions. One is that looking at drivers that have led to the diversification of species and populations, how we can use omic tools to look at not just biodiversity, but adaptation and human health and ecology. This is primarily through the integration of eDNA, which is a genomic tool, but really it's an ecology kind of application. And then finally, how do genotype and phenotype interactions enable organisms to adapt to selective pressures? And I'm not going to have much time at all to talk about this third point probably, but I'll touch on a couple new projects that we have starting in the lab. So first, looking at this first question, the diversification of species and populations, I want to show you, how studies of evolutionary biology are more impactful when we think about these four components together. Many of you, I don't really have to introduce this group to a lot of people here, which is great. You might be familiar with the caringoids, new clade name. You can read it in our paper. The caringoidia is a group of fishes that's well known. A lot of popular fishes, we call these, mahi-mahi or dolphin fishes, the remoras, or shark suckers, the cobia, and then the most speciose group within this clade is the carangids, so the jacks, trevallies. At Seshni's talk last week, she mentioned this is a huge part of SAIAB's collection, and it's been great to be working here with the biobank in this collection. The reason I like studying carangids, not only because they're fun to catch, but because there's really important fisheries for them, whether those are for subsistence, commercial, small scale, or sport fishing. This is actually a farm of yellowtail trevally in Hawaii. And so studying this clade allows me to look at the intersection of these different types of fisheries from an evolutionary standpoint. And thinking about kind of how genomics can contribute to the sustainability and sustainable management of species within this group. This is a map of species richness of the caringoids that I made using expert range maps from IUCN and Aquamaps. And the bulk of diversity, unsurprisingly, is in the Indo-Pacific around this coral triangle area. And I just love this map, not only because it's kind of pretty, but if you think about it, I mean, if you have 120 species inhabiting this one area, yeah, there's a lot of habitat variation and things, but it makes you really think, like, why and how do species overlap in their range? What are the factors that allow them to coexist? Are the ecologies different? Or are their habitats different? Even with a group of kind of large-bodied, widespread species like carangoids, what determines the what, when, and how they're able to survive in a given place and time? And you can think about that kind of question in your favorite biodiversity hotspot or for your favorite clade, whether those are fishes or inverts or whatever. And that sort of component is a fundamental question of biology that is super important to address given the present and also given the future and how habitats and ocean conditions and freshwater are expected to change. Of course, a lot of what we understand about the present, we know from the past. And therefore, I want to say fundamentally, this comes down to evolution. And the best starting point we can get with evolution is a phylogenetic tree, a resolved phylogeny. So the carangoids had a sort of contentious taxonomic history. These are just some of the phylogenies that were generated over the years using different mitochondrial and nuclear loci, yet they were often conflicting in their topologies of not just carangoids, but their parent clade, Carangiformes. And so the objectives of my work with this clade were to construct a comprehensive phylogeny in terms of both taxonomic and genomic coverage, to integrate fossil calibrations so that we can accurately estimate the ages of member lineages, which then we can use to reconstruct and infer patterns of biogeography and their kind of range, expansion, and history. And so this is work that was just published in November. Again, taking this theme of biodiversity, integrating tools like phylogenomic and large genomic data sets with species ranges to answer questions that are important for conservation and management related to species distributions, resource competition, and taxonomy. I don't have time to go through all of the real method-y details, but I'll say that we use a method called ultra-conserved elements. These are regions of the genome that are, unsurprisingly, highly conserved. They're also abundant throughout genomes and easy to identify. And they're useful phylogenomic markers because we can look at the sort of distance from those conserved regions, which reflect mutations and evolutionary change over a given time period. So we targeted over 1,000 loci and ended up with an average of about 985, sampled 137 species of carangoids, which was 80% of the clade. And again, I had to take out the tips because this is a big phylogeny, but we were able to accurately develop the most comprehensive and well-supported phylogeny to date of this group and estimate the age of origin of this clade, as well as the ages of the member lineages. And we found some interesting patterns taxonomically as well. Specifically, many genera were in fact not true genera, but were paraphyletic, shown here by the different colors. So Carangoides, for example, is shown in blue. It actually fell out into 9 different clades. And this is important because it suggests that a taxonomic revision of Carangoideae is necessary. And in this case, the taxonomy doesn't reflect the evolutionary history of this group. And that's important because if you think about it, we're going out into the field as scientists, or maybe there's fishers or citizen scientists that are needing to use these keys and identify these species. I don't really need to push the benefit of taxonomy here at SAIAB, but it really is important. And I think this is, this clade is pretty understudied. And so if there's any morphologists in the room that, are interested, there's a great starting point here to work with. And I also want to emphasize that having a well-resolved phylogeny, for me, is as someone who's kind of a big picture thinker, is just the start. It's necessary to answer those questions that relate to the map I showed about survivability and habitat and competition. And so really thinking of what has shaped the evolutionary history of this species, right? And one way to look at that, like I mentioned, is to look at these patterns of biogeography. But in a clade of fishes like Carangoideae, this is just a subset of species, and this was a BioGeoBears ancestral range reconstruction done by Alex Sequeira. He was at JCU, he just moved somewhere in Western Australia. This kind of analysis is challenging because so many of these species are already widespread. These are their contemporary ranges here on the right. And so it doesn't take much time. These methods aren't really able to distinguish between dispersal scenarios. And they assume that most of these ancestral clades were widespread as well. So it doesn't take that much time before suddenly like any part of the ocean is a possible ancestral range, right? And so that's challenging because for years, scientists have been trying to describe barriers and biogeographic provinces for marine fishes to try to come up with, unifying theories on mechanisms of speciation in the ocean, right? The prevailing assumption is that speciation, even for marine species, is, even though the overlap in range is allopatric, meaning that speciation occurs when those lineages are separated by some sort of physical barrier. And yet, as I just showed, there's clearly challenges with these ancestral rate reconstructions to contribute to these sort of bigger picture aims of marine scientists and evolutionary biologists. And so one way we can kind of try to start to get around that is just by looking at contemporary ranges. Are there patterns we can see using their contemporary ranges that might infer speciation mechanisms or what's going on? So I looked at patterns of range overlap and range symmetry, range size for 123 species of carangoids that we had data from IUCN and Aquamaps, and identified 41 sister species pairs, meaning they're two species that are each other's closest relative. And I calculated some different metrics. So first was range overlap, whether they're allopatric or sympatric. This was calculated by the dividing the sum of an area occupied by a given species pair divided by the area of the smaller range species. And that might be a value that's ranging from 0 or no overlap all the way up to 1, where there's complete overlap. And then calculated range symmetry or range size evenness, where we took the value or the size of the smaller range species divided by the sum of both species ranges. And that's a value that's ranging from 0, where they have very different range sizes, to 0.5, where they have very equally sized ranges. And so laying it out in the sort of four sort of extreme scenarios, you would have a case where you might have low overlap and high symmetry, high overlap and high symmetry, low overlap and low symmetry, and high overlap and low symmetry, if you can see that. And in real life, it looks something like this. So here's two examples of sister species that show no overlap, and sorry, this is a little bit low, no overlap and low symmetry on the left, meaning one has a, they don't overlap in range, and one has a small range relative to the other in dark green, that's a larger range. And then high overlap and high symmetry on the right, meaning they both overlap a lot everywhere, actually, where it's hard to see in this figure, but everywhere there's dark green, there's also light green. So they overlap in those areas, and then they have equally sized ranges. You can also look at how those relationships differ based on node age, so the time of divergence of that node that contends those two species. And so that's what I looked at with all those 41 sister species pairs. So this is our results, and what you see here on the y-axis is range overlap, so ranging from 0 to 1, and node age, so that age of divergence on the x-axis. And every point here that's black is an allopatric pair, and every point that's sympatric or is denoted in blue here. And what we found was that 73% of sister species are sympatric, so 30 out of 41 pairs. I'll say that no level of symmetry has been observed before in the large group of marine fishes before this. And now there's been a study on myctophids that found something similar, but generally speaking, this is for a widespread, pretty large clade like Carangoidea. This is a big amount of symmetry. And I want to point out too that in most cases, this overlap is quite high, like 60 to 100% of the ranges that they're overlapping in. So this plot shows range symmetry on the y-axis, so that range size evenness, and range overlap on the x-axis. And we didn't see super strong patterns. I'll say too that with node age, we didn't really find anything that could infer a mechanism of speciation. With symmetry as well, we didn't find any sort of strong pattern, but most of the points are here in this top right quadrant, indicating pretty high overlap and high symmetry, so even sized ranges. And remember, oh, I want to say that for those 11 spares, or sorry, 7 out of 11 pairs that were allopatric, 7 out of those, so there were 11 pairs of sister species that were allopatric. Out of those, 64% had the exact same distribution, where one species was found on the eastern coast of North America, and the other, or this is the western coast, sorry, the eastern Pacific, and one in the western Atlantic. And those pairs diverged around 5 or less than 5 million years ago. And we know that the Isthmus of Panama diverged between 3 to 5 million years ago. And I just did the math this morning, and not to like discredit the Isthmus of Panama, but if that wasn't there, that would mean that 90% of the species were sympatric, right? It's just that, you know, I mean, obviously this is important, and it was important to the divergence of these species, but it's just amazing how much symmetry we found. The other cases of allopatry tend to be due to like open ocean barriers, like shown here across the Atlantic, or even the influence of the Benguela and Agulhas current. We found some splits there as well. But again, 73% of sister species were sympatric. And you might think, you know, species that are each other's closest relatives, if they overlap so much, how are they able to maintain, how are they able to stay separate species, right? And this is a 2D, right? This is a 2D allopatric view or lens. And so thinking, right, there might be something else, like symmetry could be in other dimensions. Allopatry could be in other dimensions. And so we were interested in looking at ecological traits and habitat traits to see if that had any impact as well. And indeed it did. So what we looked at was for every sister species pair, we compared allopatric or non-overlapping with sympatric sister species pairs, looking at differences in depth that they're found, maximum depth they're found in the water column. And that maximum depth was mostly due to limited data for kind of depth ranges. And then maximum body length. And we found that there were greater differences in water column depth and body length to some extent in sympatric pairs compared to allopatric pairs. Okay, so greater differences. And these results are indicative of ecological partitioning, right? Allowing sympatric species to remain distinct lineages despite an extensive amount of overlap in their range. Digging a bit deeper, we also then just looked at sympatric species, and we found that it was also influenced by habitat type. So when 2 of those sister species pairs occupied the same habitat, meaning either both were reef associated or both were non-reef associated, they had greater differences in their water column depth significantly than sympatric species where one was reef associated and one was not reef associated. And so this shows even more strongly that ecological partitioning is occurring. And so broadly speaking, you know, understanding why species overlap and why they don't, and a perspective that's driven by evolutionary processes is leads us to new questions. It leads me to thinking about new questions. And one thing I'd love to do is to start, gathering samples from individuals where they do and they don't overlap in their range to really test if true, character partitioning, character displacement is happening here. And I want to emphasize too that, having the genomic, I was lucky to have this great genomic data set that can lead to these big picture questions. And so I think about, from the phylogeny, then I look at, okay, well, let's tease out these individual lineages, looking at how many populations of those lineages are there. And do those populations show similar, species population level responses to stressors across their ranges? And putting that kind of question here in a phylogenetic context is really interesting to me when you think about kind of shared adaptation across clades and the ability of different lineages to adapt or not adapt to different changes in the environment. So I want to then just touch on this population connectivity part, and I kept this in here because I know kingfish, I call them trevallies, they're the same. I know they're so deep to so many people's hearts here in SAIAB and in South Africa. And so I was really interested in this theme of population connectivity, which we could understand with these great genomic data sets, as well as efforts of citizen scientists to look at these management implications on stock structure, genetic diversity, and anthropogenic stressors. And I focused on 2 species, the bluefin kingfish and the giant kingfish. They're not each other's sister species, but they are known to hybridize, and they diverged about 5 million years ago. These species are iconic sport fishes, right? This is, you might recognize some of these guys. They're found all, they're generating a lot of money and interest, and not just, economic interest, but conservation interest across their ranges. People are paying thousands of dollars to go and fish for these across the world. And also importantly, if not more importantly, they're in Hawaii and other Pacific Island regions like Tahiti and Samoa. There's a very unique cultural history behind fishing for these species, including traditional fishing methods, language names, stories, and components of folklore that are tailored specifically around giant and bluefin kingfish. And this is important because in Hawaii, there's a lot of conflict over resource allocation and overfishing and indigenous rights to their fisheries. There's still a lot of fundamental biological questions that aren't answered that we need to understand to be able to practice conservation in a just way, right, in an equitable way. In Hawaii, they're mostly unmonitored and unmanaged. We also know through work that I did with Ryan Daly and Paul Cowley and others that giant trevallies are apex predators. We did a study looking at stable isotopes using compound-specific acid stable isotopes looking at different sites, 1 in Mozambique and 3 in the Seychelles, that represented different habitat types, and found that in some cases, these are adults. Not only did we find differences in their trophic niche space based on habitat type and location, but that these are really top predators. I mean, they have some similar trophic positions here as bluefin tunas and some of these really top predator sharks. So ecologically, these species are, of course, very important. When I started this work, this is the range map of bluefin and giant kingfish. So blue is shown in blue and GTs are shown in red, and where that they overlap is in purple. So they overlap throughout most of their ranges. It's kind of hard to see here, but there's just only a couple areas where they don't overlap. And one question that was unaddressed was the genetic connectivity and diversity of these species. And when I started this work, there was only one study that had been done in Hawaii. And so given that Hawaii is just one component of their ranges, we conducted a global population genetic study of these two species, looking at the extent of connectivity across their ranges, and particularly looking to see if there was geographic structure. If anyone's ever tried to fish for GTs and isn't used to it, I guess, like I am, it's quite hard. Even if you are used to it, it's hard to fish for these things, and they're found all over the place. So how do you get samples for something like this, right? We worked a lot with sport fishers, and this was a key component to getting this project done. I relied on collaborations with sport fishers from 13 different countries. We sent little sampling kits, organized the permits. Some got really excited about it. They put it on their social media. Their clients helped them collect fin clips. And so this was a really kind of fruitful and important part of doing this work. Also to kind of learn their knowledge about species behavior. So in the end, we gathered hundreds of samples of both species, just a subset are shown here that we included in our final analysis. And these were collected from 13 different localities across the Indo-Pacific. While I was doing this work, developed new collaborations with Brandon Pickett and Keone Coway. Brandon is now at the NIH, amazing bioinformatics wizard. And he, for his PhD work, we generated the first reference genomes of these two species. And so those genomes are publicly available on NCBI, and it's been really fun to work with them. We were able to map our RAD-seq SNP data and generate tens of thousands of SNPs for this analysis. So what we found was that for bluefin trevallies, we found evidence of stock structure. And I want to just point out, this is a PCA, so points that are closer to each other are more genetically similar than points that are farther away. These ellipses don't represent statistical significance or variance or anything. I just drew them because it was easier to kind of show these regions that we're seeing. Same with giant kingfish. We find that there's evidence of stock structure across their ranges. And I'll show another map here for bluefin trevally. This is showing, this is an individual ancestry plot that we generated using structure. So each bar represents an individual fish. And essentially this method uses an admixture of it's a model-based clustering analysis. And so it uses a matrix of genotypes to estimate individual ancestor coefficients. And so for the bluefins, we found, a putative separate population in Hawaii. You can see that there's some, we could say, infiltration of other individuals that kind of have ended up in Hawaii. And there's some reasons, I think, for that based on the patterns of currents that I don't really have time to get into. And then for the giant trevally, similarly, we found this separate population shown here in the blue in Hawaii, but also in the island of Kiribati, again, with some admixture coming in from this Western Indian Ocean clade. And I want to show, it's really evident if you look at a pairwise heat map of FSTs, of genetic differentiation between these regions there's a lot of different differentiation from down there at the bottom, but these represent Hawaii and Kiribati compared to everything else. And then pointing out that there's really very, very, very low, a lot of gene flow happening in the Western Indian Ocean for these. So, thinking about other work that people are doing here and connectivity of these species, it's interesting. One thing I'd like to do in the future is sort of narrow in and try to figure out, well, where are those biogeographic breaks occurring? Where are the spawning populations? We know about a couple, right? But there's most of them that we don't really know about. And I want to just kind of drive home that this is important because, for example, in Hawaii, giant trevallies are overfished, and overfishing is continuing to occur. According to NOAA's estimates of annual fishing mortality rate as well as spawning potential ratio. And this is more concerning in Hawaii because of the reasons I mentioned earlier on indigenous kind of practices for these fishes, but also because this is a federal assessment, and most of the fishing occurs in state waters where these fishes are unmonitored and there's really not any fishing regulations. It's worrying because they're top predators as well. So we know what happens when you take top predators out of the environment, and it's not pretty. And so, yeah, I'm excited to keep asking more questions with this work, and I want to just quickly pivot to, I've got more to talk about, but I want to pivot to some of the work that, very briefly, that we're doing in Alaska, where bigger isn't always better. I'm going to say that, but it is cool what you can learn from bigger genomic data sets. And we can really ask some of these bigger picture questions. And so this is, it's so funny. I get to orient people to the Bering Sea, and we get so siloed in Alaska. So let's travel across the Indian Ocean up to the North Pacific. Here's Alaska. We have the Bering Sea here in the Gulf of Alaska, and herring may not be as fun to catch as GTs, but they're just as important for cultural purposes, for economic purposes, for ecological purposes as forage fishes. This is work that's been done by my master's student, Sydney Almgren, and my postdoc, Laura Tim. And we've known for a while that herring in the Bering Sea from mitochondrial data are different than herring in the Gulf of Alaska. So if you catch a herring over here, it's actually more closely related to a herring from Japan or Russia or Korea than it is to one in the other part of Alaska, which there's this biogeographic break here along the Aleutian Islands and the Aleutian Trench. And so most of the studies to date, if not all of them, actually have, well, in the Bering Sea have used mitochondrial data sets. But we were like, let's go big. So we did low coverage, whole genome sequencing. We got the full genomes of these, but at not a super high coverage. And we were able to map these to the Atlantic herring reference genome. They're very similar. Ended up with, in the nuclear data set, 4.7 million SNPs. So compared to even 40,000, this is a huge amount of data, plus 300 SNPs from the mitochondrial genome. And what we're finding is that while there's two populations in the nuclear data, the mitochondrial data actually suggests three populations: the split between the Bering Sea and the Gulf of Alaska, as well as two populations that are co-inhabiting. These individuals were collected from the same exact location at the same exact time. And I'll talk about that in just a second. So one thing that's cool about these low coverage whole genome data sets is that you can look for kind of where that genomic signal, where that divergence is occurring in the genome. And we're interested in like what people call structural variation. Are there inversions? Are there large sections of the genome that aren't undergoing recombination that might be driving those differences? And so that's, if you lay out the whole genome, and separate it by chromosome, this is if you think about it, if you found localized signal, you would expect like maybe a peak of strong differentiation at some point in the genome. So SNPs that are really, really highly differentiated between individuals or populations. That's what you might expect if you find structural variation. And what we found was that no, it's just, these are FST values. It's all, the whole nuclear genome is just like between the Gulf of Alaska and the Bering Sea is very different. It's not just one chromosome that's driving this. It's just, these are huge FST values. They're actually higher than values that a recent study showed where they looked at Pacific herring and Atlantic hybrids. So massive differentiation here. And I want to just say quickly that just last night, Laura was texting me on Slack about the differentiation in the mitochondrial genome, and that most of the SNPs that are different are found in gene coding regions that are in the NADH2 region. So there's NHG2 1 through 6. Those are responsible for this complex 1 enzyme complex. And there's a study that showed similar results from stickleback. Then they inferred that maybe that was from a relic hybrid population. So it's really cool to be able to go into the genome and actually see, well, what genes are responsible for those differences. And if that complex 1, there's a lot of work on humans with that complex that can lead to mutations in that region, can lead to diseases, and the region is responsible for producing ATP energy. So we're going to still keep delving in to see, okay, well, why are species that are in the exact same place at the exact same time, why are these two mitochondrial lineages able to coexist, and what does that mean for the health of these herring? Okay, I'm going to pivot here. We got some time. We're going to change focus a bit, and it's just going to be exciting. I brought a prop, so get ready. We're going to talk about how omic tools can link species biodiversity, adaptation, and human health. I'm going to briefly introduce a project looking at glacial retreat on biological communities. This is the coastline of the Gulf of Alaska, or part of it. We are a glacial dominated ecosystem, and those glaciers are rapidly changing. And so one thing that we're interested in is how can we monitor that in a cost-effective way? I'm so excited to show you this figure. Most people, when you see Alaska, I think a lot of people think it's part of Canada, which we wish we were sometimes, but we're not. The coastline of Alaska, this is Alaska overlaid across the rest of the US. You always see it up here where it is, and it looks so small. It's massive. It's a massive state. Our coastline is greater than the entire United States coastline combined, right? And so thinking about these processes, especially the impacts of climate change on coastal communities, we know that climate change is accelerating at 4 times the rate in the Arctic than anywhere else, except maybe the Antarctic. And so it's really important to think about like I said, biodiversity monitoring and how we can use omic tools. So this is a project that was started by my master's student, Maris. When I moved to Alaska in 2021, there were folks that were answering these kinds of questions of how, as glaciers recede, how do communities change? And they were looking at fishes, mostly using beach seining. And you can see, I mean, we know that these types of traditional fishing monitoring methods are useful, but they're also expensive and they're limited, like any tool, right? You don't use a hammer to screw in a nail. So these guys with their beach seine, they're not catching fish over here. They're not catching fish smaller than the net mesh size. They're not catching invertebrates, and maybe, hopefully not mammals or seabirds as well. And so we thought, okay, well, let's use eDNA to compare and see if we're finding the same kind of patterns. Again, Maris has collected a ton of samples. She's analyzing those now. We're getting some cool patterns. I'm really excited to see the invert patterns. But it just leads to, this project has led to another goal in my lab that relates just to the use of eDNA in general and as a biomonitoring tool. And so I see one of my goals in Alaska where eDNA, just like here, is still relatively new. Like people are jumping on the bandwagon, but it's not like a super established method, at least in fisheries, right, and in monitoring. And so I would like eDNA to be more affordable. I would like it to have a whole variety of applications, and it has to also be user-friendly. We're a very rural place. Just like here, taking the pumps and everything with you into the field can be expensive. My poor students trying to filter that glacial silt water, it took hours. And so normally what you think about when eDNA sampling is what I would call active or conventional sampling, where you're going out and you're taking a bottle of water, maybe you're taking a CTD cast on an oceanographic vessel. There's some fancy backpack samplers, but this technology is expensive. And I'm not to bash Smithford here, but I mean, the citizen science sampler that they offer is $1,800. I don't want to pay that much. I don't, just for a citizen science sampler, that doesn't even include the price of the filters. This is an auto sampler that they sell. It takes 8 samples. It's $28,000. The cheapest auto sampler I found so far is $280, but still that can get expensive, and it's hard to make something like that. This is like the, I don't know, Lamborghini or Porsche of auto samplers. It's $55,000. It can take 9 samples. It can also kind of preserve samples in real time. It has a great application for deep sea research. It was developed by folks at Dalhousie. It's just really expensive. I think hopefully it makes you coffee at the same time. And yet that it's a limiting factor, right, for cost-effective biodiversity monitoring. They took clothespins and they put filtered fence posts, they put it in the water, and they found just as good results as taking conventional methods. And I was looking at this, and we just gotten a grant for like $50,000 to develop this passive sampling technology, and it was leaking, it wasn't working. And I was like, why would I spend all this time and money doing this fancy stuff when we can just do this? Also, passive sampling has a merit in that you can look at DNA, you can look at patterns over a given time period, overnight, over a tidal cycle. And there's some more and more studies that are coming out now that are focusing on this. And so we've been exploring passive sampling technology, so to speak, in our lab. Maris, my student, did a little study. We're also wanting to compare different types of filter types. So like glass fiber filter, nitrocellulose. We put Amazon sterile gauze that we bought from Amazon for $20 out into the field, and we found 12 times the amount of DNA as these filters that we're getting from Thermo Fisher from the companies. That was in a highly sedimented environment. We're not finding the same thing in low sediment environments, but we're still getting plenty of DNA from this type of filter, so to speak. But probably in this case, of course, there's a large pore size, there's a lot of sediment. And they took this capstone course, and they were tasked with I tasked them with a challenge, so to speak, of designing a device that aligns with the currents and also functions as a sediment trap. And so we went back and forth over the academic year, and there we go. Tested out different prototypes. What would this look like? I would come in and wave my hands and be like, let's do all this stuff, but I don't know anything about engineering. And then they would draw a draft, and then it would be great. So this is what we came up with. It's called PESCA, Passive eDNA and Sediment Collection Apparatus. And I'll show you here. I actually brought some. So what happens is that water will come through the center here. It's aligned with currents by this T-mount where you can attach it to a buoy or a dock or even tow it, allowing this to rotate with the currents. Water comes through, it hits this dam at the back, which creates an eddy, and then sediment now drops, this one's a little bit tight, drops into this little cup here. And so not a lot of sediment, but you can compare eDNA that's on the sediment with eDNA that's on the filter. You can look at mercury content, you can look at, I'm not gonna put it back. Different questions, and then you have a cap here at the end that could hold regular filters or sterile gauze is what we've been trying out with. And so we've been piloting this technology. Oh, and the best part is, anyone want to guess how much it costs?
$50.
$50. Anyone else? 50 cents. Oh, okay, anyone in between? $10. Okay. $4, everyone, $4. So yeah, anyway, we want this to be cost effective, right? And material costs. And it's 3D printed. I think I said that, I don't remember. So the goal is to kind of see, well, what questions can we ask using something like this? Is it comparable? Are there limitations to using a device like this, and what are the benefits as well? So we were able to test this out last summer with 2 of my undergraduates at one of our field stations. I didn't get my boat safety training done in time. So at the last minute, I said, we're going to buy kayaks. So we did carbon-free sampling with kayaks out in the bay. We deployed these buoys and we were just trying to see, like, does this behave like what we want it to, or the way we want it to? So we were comparing it with other types of little 3D printed devices. We had railroad ties at the bottom, a line that fed up, and then we were also taking conventional samples at the same time to see, okay, are we finding different patterns in community composition? So there it is, sort of suspended in the water, behaving like we want it to. Some folks in Hawaii took one of these out, it's upside down there, I hope it righted it, to attach to a glider earlier this year. And I'm always looking for partners. So if you're doing eDNA work and you want to try these out, I have funding to print more and send them to people. One thing that we found is that eDNA, the concentration increased with soak time. So we tested this at 3, 6, 12, and 24 hours, and not longer than that. So there's still more questions of how long could you leave this out until you're seeing this sort of saturation in what you're able to detect. Our library that we submitted, which was the 12S MyFish library, those results, the sequencing results, like just came back like 2 days ago. They're sitting in my cluster waiting to be analyzed. So I don't have results on that, but I think that there's a lot of really cool applications, of course, that we know for eDNA in general, but something like passive eDNA sampling could help better answer some of the applications, like detecting invasive species, right, or diseases and applications that we come up with as we bring new minds to the table and explore new places. And one, very quickly, I want to just highlight a couple of projects where we're using passive sampling. This one is brand new. It's just funded by NSF, part of a big $20 million 5-year initiative to look at the impacts of climate change on coastal communities, and particularly mariculture. This is a partnership with Chugach Regional Resource Commission, which is a consortium of 7 Alaska Native tribes. They have an amazing research institute called Ecological Pride, where they have hatchery, they raise clams and indigenous food species, so sea cucumbers and crabs and cockles and things like that. And so we're actually going to work with them to try to see if we can use eRNA to distinguish larval and adult clams in the wild, in water, to see, okay, well, where are clams spawning? Because there's been some declines in native clam populations. So I'm excited. Next time I present here, I'll have so much more information. It'll be very exciting. And then I want to highlight a really cool study by one of my undergrads, Emily Entrikin. Emily is a professional rafting guide. She came back to get her bachelor's after 20 years of guiding in Arctic rivers, and she wanted to have a science question, and passive sampling with these 3D printed devices is really amenable to citizen science. So this is a picture of the Firth River. You can see, maybe not, there's these tiny people, including Emily. And what she did was put out, she's getting paid to take clients down these rivers for 10 to 12 days at a time. And so last summer she went to 3 Arctic rivers, and the question she was trying to answer was, can we detect salmon, Pacific salmon? Because salmon were always, for a long time, been known to kind of inhabit and move sort of into the Arctic waters, but only until recently, with water temperatures changing so quickly, are salmon starting to spawn and colonize, so to speak, in the Arctic. And that has a lot of implications because there's other important subsistence species there, like whitefish or sheefish, that are really important to communities there. And salmon are ecosystem engineers, right? That could be a huge impact. Whether it's good or bad remains to be determined. But they're starting to move in. Another colleague of mine found a spawning population just last year doing field work. But if you think about an effort to go down here, if you were to organize a scientific expedition, right, with the helicopter and the gear and everything, that's going to cost tens of thousands of dollars. And Emily did this by printing these things for $4 and taking her clients and putting them out overnight, in addition to traps. And it's a little bit, don't worry about the map, but basically we are finding both pink and chum salmon positive detections at some of these sites. We didn't look at every site just due to timing and cost, but I think she wants to go back this year, and it's just a really fun example of students being able to integrate this with work that they're doing that they're passionate about. Okay, and I don't really have time to touch on this last part, but I want to say that we have a really cool project that's starting looking at genotype-phenotype interactions in cold environment. Now that I live in Alaska, it's very cold, and We're looking at the evolution of cold tolerance and intertidal invertebrates, specifically by looking at their ability to produce ice-binding proteins and kind of characterizing how these animals, even though they're technically freeze tolerant, how they're able to prevent damage to their cells that occurs with ice crystals as they're in the intertidal, and you have this very cold air, water interface. There's a sea urchin and a Pycnopodia sea star. So again, next time I get back to SAIAB, we'll be able to chat about that. But in the end, I wanted just to wrap up by saying, I hope that I demonstrated today through these examples of collaborative projects, the importance of the integration of, I didn't really talk about the tagging behavior work, but genotype, phenotype, and behavior and environment, and how we can integrate the multiple big data types to really answer these big picture questions that have real relevance for conservation and management. I want to say thank you to all the sport fishers and citizen scientists that have helped me throughout the years on the Carangid work, as well as a lot of funding sources that have also helped. A huge thanks to SAIAB, the collections team here, for helping me throughout that work in my PhD and postdoc. And even currently, I think we were asking Sesh for samples the other day. And thanks to you all for listening. I'll go back to this page in a second. Really, all of my students won awards at the American Fisheries Society Alaska Chapter Conference. I was the most proud PI ever. And I want to just quickly say that tomorrow morning, I'm giving a CV writing workshop in this lecture hall from 8 to 10:30. So please come and find me. You're lucky sent around a link. So please, if you're interested in doing that, even virtually, click on the link and register and come and bring your CV, and we can exchange and chat about that. So students, interns, staff, you don't have to, anyone can come. And with that, I'll end it and say thanks and take any questions.
Well, thank you very much, Jess. Very cool talk. Yeah, a lot of nifty projects on the go at the moment. And like we were speaking about yesterday, we've got a project on looking at different behaviors and the underlying sort of genetics behind that. So yeah, some very cool stuff. Are there any questions from the audience first before we jump online? Yeah, Wink.
Yeah, thanks, Jess. Yeah, great talk. In terms of the beginning part of your talk, you spoke about sort of the allopathic separations of some of the Karangas, you know, in Panama, and then obviously you can see the Red Sea as I've always wondered what, you know, is there much now that those are open? Well, maybe not Panama so much, because there's obviously a lot of fresh water in terms of the Panama Canal, but like in the Suez Canal, you know, any species that wouldn't have been connected past, but are sort of being anthropogenically connected.
Yeah, there is. There's a few years ago, I read a paper about like even some Karangids that were moving into the Red Sea that hadn't been there before. Yeah. So I don't think Panama, I don't know, actually, maybe certainly like, yeah, certainly invasives and stuff, but yeah, there are instances, and I haven't looked at it in a while, but I know there is like colonization, or there are colonization events occurring in the Red Sea when that canal's opened up. Yeah,
and even a hybrid zone almost going on there, potentially with, you know, but I mean, a lot of the Mediterranean species, the southern Mediterranean species, they're a bit less in terms of temperature, but yeah, just curious on whether that area is becoming more interesting the longer since the Suez has been open. You know, how are the species, especially the allopatric species that were split, and now they're together, are they hybridizing? Are they ignoring each other? They speak different languages?
That sounds like a good research proposal. Yeah, I am not super clued up on like specifics, but I know that I've seen papers that I've shown like, you know, detection of some Korean, maybe some of the scads and things in that area. Yeah, good question. Yeah. Francesca?
Thanks, Jess. I love finally getting to the larval dispersal application of the eDNA or pre-RNA. I wanted to ask you something about your eDNA minion. I've seen the deployment that you showed, quite sheltered waters. What do you think, or how would that apparatus behave in quite dynamic at times in rocky shores, or like very, very close to shore type of environment? And because you measure sediment, I guess you've got some timing that you need to deploy, but could you think about deployment over slightly longer time frames?
Yeah, those are all great questions. you know, for the first one with the really dynamic shoreline, I mean, I wouldn't put these, I did drop one this morning and it didn't break or anything, so that was a good sign, but I wouldn't put this something where it's gonna be like, you know, bashed up against the rocks. Like it needs to be able to, and obviously it can align with currents, but if it's getting flipped over, you know, that's gonna defeat the purpose of that sediment cup. That longevity question, and to your point, that's a question like the sort of application of this in a really dynamic environment is something that I would love to test or have someone else to test and see how it works for them. So TBD is the answer to that. In terms of longevity, I also don't know the answer. I wish we had set them out for more than 24 hours, but we didn't. When I get those data back, and if we see, you know, saturation over that time, that's going to be really interesting to say, okay, well, how long can you put this out? Is it just 8 hours? It might also depend on the type of filter that you're using, right? And the amount of sediment in the environment to see if that's getting clogged or washed off or things like that. So I wouldn't put this out for a week, you know, that's not being preserved either. There's no preservative here, but yeah, we want to start pushing the boundary of kind of how long we deploy these and seeing how that plays out in terms of species composition and saturation. Yeah, good questions. Yeah, back.
If I remember correctly, also like the distribution for Carangids were like northern subtropics to southern subtropics. And if I remember correctly, you're saying that there's less genetic mixing in the Western Indian Ocean.
For giant trevally and bluefin trevally, correct. Yeah.
You think that's just because of our topography or not optimal distribution, like habitats?
I think my first go-to would be like larval distribution. It doesn't take that much mixing for panmixia, so to speak, to occur. And given the currents in the system, that would be my guess. There's a lot of really cool work you could do on like source synth dynamics there still with data, but yeah, they're not, I mean, we know that they're migrating, right? But like hundreds of kilometers, but not thousands likely, or maybe we just don't know yet, thousands of kilometers. So what I would love to see is like, where are they spawning in Seychelles? Where are they spawning in the Red Sea or Madagascar? And with that information, we can then map, you know, larval connectivity and see, okay, is that actually what's driving this? Which is what my first guess would be larval distribution. Yeah. And those are just two species, right, of several more that we could look at those patterns across this area. Yeah.
Anyone else? Yes, Rick.
Thanks, Jess. That was a fantastic talk. eDNA and the expense, you can manually collect it and take time. Obviously, that's cheap. But the limiting factor for a lot of the work that we are trying to do is the cost of the analysis, obviously. So is any work being done to reduce the cost there, in addition to reducing collection costs?
That's a good question, right? And that's like, it's not just the analysis, it's the bioinformatics expertise and the like sequencing costs. This doesn't, like you said, this can maybe reduce those upfront costs. It doesn't help with that. My best answer is like some new kind of kits that allow you to more rapidly go from collection to like library prep to sequencing, but still it's sequencing costs at least are coming down. I know there's been some cases like where you can take the MinION out, you can do metabar coding with a MinION and get data in real time. So costs are coming down, but they're still expensive, and that's still a limiting factor, especially here. So I don't have a good answer to that part, but at least you're saving hopefully hundreds of dollars at the front by not having one of these backpack, if not thousands of dollars, backpack samplers and getting cheaper filters and things like that. Yeah.
Do you want to just get the comments there?
Someone asked, was lineage sorting used to explore divergence in the GT, like incomplete lineage sorting? No, I didn't do any analyses of incomplete lineage sorting there. One thing I would love to look at in more detail is we did find some evidence of hybridization. So that is something I'd like to keep exploring, but no, we just did, what we did was the PCA work. We did the SNMF for structure analysis, and then we looked at pairwise FSTs. We looked at haplotype diversity, genetic diversity, but we didn't do that's the extent to which of analyses that we did for that work. I hope I answered your questions, Sven. Feel free to unmute or respond in the chat.
Anything else? Thank you, from Sven.
Yeah.
Okay, I think with that, Jess, we'll end it there. Thank you very much for taking the time to give the talk, and thank you everyone for listening. Thanks, Jess.
Yeah, thank you.
I'm keen to